Yes. So today our Kipoi seminar is with Noor Youssef, so you're working in the lab of Debbie Marks, and will present work on viral evolution and therapeutic design. So Noor, thanks. And without further ado, the stage is yours.
Awesome, thank you. You can hear me okay? Just checking.
Yes.
You can hear me? Great. Awesome, thank you for the introduction. I'm excited about our work, how we are predicting how viruses evolve and mutate and change over time, and how we're using that to design vaccines and other therapies with an eye towards that future evolution. So, I mean, this statement is probably obvious to everyone. Events of recent history, the last 6 years, viruses have had an enormous impact on humanity. It's one of the most unifying human experiences that every single one of us on this Zoom has felt at some point, is what it's like to be home sick with the cold or the flu. And for most of us, that means a day or two of feeling under the weather, but in a week or so, you feel totally fine and you forget that you ever had a viral infection. Now, that story can be very different depending on the virus that you were infected with. Just a few weeks ago, I was talking to a complete stranger at a coffee shop, and it turns out that her father had been permanently paralyzed from the waist down because when he was a child, he'd been infected with a polio virus and he hadn't been vaccinated. And it ultimately led to paralysis, and how he, decades and decades later, ended up passing away from pneumonia, an infection in the lung that he likely could have overcome had his lungs not been compromised by this virus that he had decades and decades ago. And so those two examples of the common cold or the seasonal flu and polio are really on the two extreme ends of the impacts that viruses can have. And so a lot of the work that we do in the lab is focused on figuring out how we can build a more proactive way in which we respond to viral threats. And most of the work that I'll tell you about today is focused on the area of how we can predict how viruses spread between people and how we can use that information to design better vaccines. And so I'm going to tell you about 3 different steps that we will get us to designing better vaccines. The first is how we can use machine learning. And really, the work that I'll tell you about today is how we are using computation, machine learning tools to forecast how viruses evolve and mutate, and how we're using that to also predict how our immune system responds to those changes, and then ultimately using both those 2 pieces of information to design better vaccines. Okay, so starting with the first step, forecasting how viruses evolve. In order to do this, we must first learn what the rules are of what makes a natural protein work or not work. And there's a variety of ways in which you can do this. The first is you could reason through it, you could think about it. And this isn't meant to be a facetious comment. Like, you can actually reason fairly well about which mutations make a protein work or not work. And the reason for that is proteins, which are made up of the 20 canonical amino acids, which are represented here, can be grouped into different categories based on their physical or chemical properties, if they're positively charged or negatively charged. But if you even take a more further out view of this, you can group amino acids in terms of their hydrophobicity, or if they're hydrophobic or hydrophilic. And one pattern or rule that comes out almost immediately is, if you look at a 3-dimensional structure of a protein, the inside of the protein is almost always decorated with hydrophobic amino acids. And then the outside of the protein is almost always decorated with hydrophilic amino acids, those that are exposed to the solvent. So that's one pattern or rule of what's needed to make a functional protein. But of course, there are other rules out there, because you can have a different protein that has an identical 3-dimensional structure, but you can have a completely different string of amino acids that still satisfy that one rule of hydrophobic on the inside, hydrophilic on the outside. But just because you have these 2 protein examples and you know this one simple rule, you can't now start making or inferring what makes a functional protein, because just a simple swap between these 2, you end up going from having 2 functional working proteins to now having 2 non-functional or broken proteins.
And so there's some other rules beyond what we can reason through. And one way that we can begin to uncover those is to do experiments. So you have a protein that you're interested in, you can create a mutant of that protein in the lab, and then you can measure the effect on some phenotype that you're interested in, whether that's the stability of the protein, whether that is its ability to bind to another protein, or the ability to infect a human cell line. And we now have the ability to not just measure one mutation at a time. We can now measure hundreds and thousands of mutations at a time. And one experiment that I'll mention again in the end, so I'm going to just introduce it now, is referred to as a deep mutational scan, where you have a protein that you're interested in, and then you create every possible amino acid mutation at every possible position in the protein, and you measure the effect on some phenotype that you're interested in. And so these are two ways in which we can learn the rules of natural proteins, but what a lot of the work in our lab and what I think is of interest to the group here is how we can do this computationally. And so there's a variety of ways in which we can use computational tools to learn from available data or sequences what the rules are of making natural proteins. And so the first and the simplest way of doing this is, if you have a protein that you're interested in, you can retrieve all homologous sequences to that protein. You can create a multiple sequence alignment, and you can look down a specific column or a position in that protein and say, during the evolution of this particular protein, how often was that residue mutated? How often, which type of amino acids were present? And you can gather a lot of information that way. But as I'm sure a lot of people are aware, if you treat every position as independent, you can run into certain issues, because of course, positions in a natural protein interact with each other, and there's higher order dependencies. And so one example illustration of this is if you have someone's name and you treat each position in this person's name as independent, and you say, okay, well, what's the most popular first name? That would be Muhammad. What's the most popular middle name? That would be James. What's the most popular last name? Wang. And so even though each of these names is the most popular at that position, the chances that I would go out in the street today and find someone called Muhammad James Wang is probably zero. So there's context dependencies, and sequence context matters. This in terms of proteins is referred to as epistatic interactions. And so we can begin to account for epistatic interactions by looking at pairs of positions, and just adding this simple additional layer of complexity opens up a whole new field. So a lot of AlphaFold, at least the initial iterations of it, were under this assumption of, if you look at pairs of co-evolving positions, you can then predict which ones are in contact in the three-dimensional structure. But of course, proteins are not restricted to two-way interactions. There's three-way and four-way and five-way interactions. But for even relatively small proteins, the number of parameters you would need to model every possible three-way or four-way interactions quickly becomes impossible to do. So that's where machine learning and computational tools can be very valuable, because they can extract these higher order dependencies without us having to write down every possible pairwise interactions. So all of these methods that I've shown so far rely as input on a multiple sequence alignment. So these are homologous proteins that are aligned. Now, for viruses, getting a reliable alignment can be challenging, because viral proteins mutate very quickly, and they also have insertions and deletions, which can make getting a reliable MSA hard. And so there's a new class of models referred to as large language models applied to protein sequences. So these are ones where we train a transformer-based model on protein sequences instead of natural language. And their main appeal is twofold. One is they're alignment free. You don't have to align the proteins. And the second is you can train them on the entirety of the protein universe, which is all possible proteins that we have access to, not just a particular protein family that you're interested in. So these are three ways in which we can learn the rules of natural proteins. And here we want to apply them to know, can we predict how viruses evolve, or is viral evolution predictable? And so I'll first focus on the H5N1 avian flu. So in April of 2025, so almost exactly a year ago, I had sent a text in my family WhatsApp group saying that I went to get some eggs and there were $15 for a dozen of eggs. And my brother-in-law responds avian influenza. Now, he's not a virologist, he's not a biologist, he's never taken a virology or biology class in his life, but he knew avian influenza because a few months before, there was the first ever human infected with avian influenza in Canada. And then a few months before that, the virus was spreading wild across the United States with over almost 70 humans who had been infected with avian influenza. And before that, it was spreading like wildfire in farm animals and in wild birds. And it led to the culling of more than 120 million different turkeys and chickens. And so what we wanted to know is if we look at the strain that led to this big outbreak, and if we train models on all the sequences available prior to this, could we predict the evolution of the virus that happened over the next 2 years? And so first, we're going to evaluate how well we do this by asking how many of the predicted mutations by the different approaches are seen over time. So the y-axis here is the mutations, the x-axis is time. And if we just calibrate expectations to what's expected in advance, if we just randomly choose positions and mutations, we would get it right about 10% of the time. If we see how well the deep mutational scanning experiments that I talked about earlier, we can see that they have a diversity of performance depending on the phenotype that they assayed, whether that was escape from antibodies or entry into different human cell lines. But we can see that at best, regardless of the phenotype, only about 30% of the mutations that were predicted to be very likely by the experiments end up being seen over the next 2 years. If we contrast that with how well computational models do, we see that they do much better. Almost 80% of the mutations that are predicted to be likely computationally end up being seen over time. So that's the first way of evaluating it. The second way in which we looked at it is asking how, so the first one was of the model predicted mutations, how many were seen? The second question is of the seen mutations, how many were predicted and at different frequencies? So on the rightmost here, these are the mutations that were the most frequent. So there were the ones that were in almost all of the subsequent strains. And we can see that the computational models are particularly well suited at identifying those very, very frequent mutations, the ones that circulate a lot. So far I've showed you how well the alignment-based methods do. If we compare them to how well they do how well the protein language models do. So each line here is the performance of one of the many language models that are out there. You can see that there's a diversity of performance, but the ones that had the best performance had issues with data leakage. And so I showed you a few slides ago where we can do the clean train test split. We can do that for alignment-based methods because we can specify which sequences go in the alignment. But for protein language models, they're on the entirety of the protein universe. And they're very expensive to train. And so once a model becomes available, we can't go back and retrain it, leaving out a specific subset. And so the issue that ends up happening is, depending on when the model was trained, some of this data that we're evaluating on was included in training. And so it doesn't really reflect how well these methods would do in a future case where we don't have issues with data leakage. And so we wanted to extend this. Imagine that today there was a brand new viral outbreak. How confident can we be in our ability to use these computational methods to predict how viruses will evolve? And so we developed a new framework called Everest in order to be able to say how reliable our ability is to predict the mutation effects of mutations using computational methods. And we came up with 2 different confidence metrics depending on the modeling type, whether it's a language model or an alignment model. And we applied this across the over 40 WHO priority RNA viruses. So these are RNA viruses that the World Health Organization has deemed of high pandemic risk for a variety of reasons, including the fact that we might not have medical countermeasures against them. And so on the x-axis, we have the confidence of alignment-based methods, and we were able to use 45 existing experimental scans to calibrate this confidence metric to be able to say where we believe we have high versus low confidence. And we can see that for alignment-based methods, we have confidence that we would be able to reliably predict the evolution of about 50% of the WHO viruses, compared to, for the language models, it's only about 7 with high confidence compared to 37 with low confidence. So that led us to two questions. Why are language models underperforming for viral proteins? And then the follow-up question is, so what? Why do we care if we can predict the mutations of on viral proteins? And so the first question is the why. Why are protein language models doing worse than the simpler old school alignment-based approaches? We were able to come up with two reasons that this is likely the case. The first is that viruses are underrepresented in training data sets. So if we look at different data sets that are used to train these protein language models, whether it's sequence-based data or structure-based data sets, they're trained on hundreds of millions of proteins. They're also trained, there's millions of viral proteins in that data set, but if you look at the percentage of those data sets that are viral, we see that viruses often make up less than 1% of training. So that's the first reason. The second thing is viruses are also disproportionately sampled in training. So if we look at all the number of all possible sequences on the y-axis, most of the time when PLMs are trained, language models are trained, you sample from the available sequences to choose which ones you will retain in training. And so on the x-axis, we have the number of sampled sequences. And each point here is a protein. In gray are the eukaryotic or prokaryotic proteins. And you can see that they fall along this line of a reduction of about 1 for every 8 non-viral sequence is retained in training. And that seems to be a consistent trend. If we see where viruses fall in this distribution, they're way far out on the left, where we have a reduction of about 1 for every 550. So for every 550 viral sequences to train, only 1 is kept in training. So those were the two reasons that we hypothesize are why protein language models today don't do well for viruses. Viruses are underrepresented in training data sets, and they're disproportionately sampled. And so those two give us indications if you were to develop a new language model tailored for viral proteins and for viral protein mutation effect prediction, you would likely address these two limitations. So that answers the why. The second question that we had is the so what? Why do we care to do this? And so I thought, I said it in the beginning, really the ultimate goal of being able to predict which mutations are viable or not is to use it to predict how viruses evolve and to ultimately use that to inform the way in which we make and develop vaccines. But another application that we did for SARS-CoV-2 is to look at the hundreds of thousands of sequences that were deposited and flag the ones that are predicted to have the highest escape. So if you look over the last 6 years during the COVID-19 pandemic, and you look at the number of samples, number of SARS-CoV-2 sequences that were deposited each month, you could see that at the peak of the pandemic, so around January 2020, there were 1 million SARS-CoV-2 that were deposited in just that month. If you compare that to this past month, that number went down to 10,000. And of course, that reduction is not because SARS-CoV-2 is no longer circulating in humans. A lot of us on this call have had SARS-CoV-2 this past winter. It's just that we're not investing as much time into sequencing the virus anymore. But regardless, whether it's 1 million or 10,000, these numbers are too big and to be able to experimentally evaluate and see what the likelihood of these variants are to be transmissible and to escape our immune system. And so what we've done during the COVID-19 pandemic is we look at all the sequences circulating in the past 2 weeks, and we score them using our computational methods. And then we flag the sequences that have the highest predicted score. And in this case, if we looked at this past 2 weeks, there was a BA3.2 variant. So in case you're not following the naming with SARS-CoV-2, this is the most recent variant of concern. It's one of the ones that's most dominant and is raising alarm bells. But this trend of the variant of concern having the highest score isn't just the case there. We've gone back to every single variant of concern that came out since the beginning of the pandemic. And if you look at the distribution of sequences that were co-circulating with it, we find that our tools are able to identify these variants that went on to become variants of concern from the very first time that virus was sequenced and the very first time that it was made publicly available. We've also showed that if we were to have used these types of methods, we would have been able to identify these variants of concern months and months before their designation by the WHO as variants of concern or variants of interest. Okay, so I've shown so far that we can use computational tools to understand which mutations are likely to retain functionality or break viral proteins, and how we can use that to predict how viruses are which mutations are likely to arise as the virus evolves. But the more important question here is, how does our immune system respond to those changes, right? If I got vaccinated with the SARS-CoV-2 or the flu vaccine last year, what are the chances that I'll still be protected this year or next year? And so we wanted to ask, not only can we predict the evolution of a virus, but can we predict the antigenic evolution, or how our immune system responds to those viruses? And so we were using SARS-CoV-2 here just because of the wide availability of data and exposure histories. So we've gone back and had samples from over 115 patients that have had diverse exposures to SARS-CoV-2. So these are people from the very beginning of the pandemic who had a wild-type Wuhan convalescent infection. This was before vaccines were available. To people who had a primary mRNA vaccine dose, so 2 doses of an mRNA vaccine, people who'd received those 2 doses and then were boosted, people who'd been vaccinated, boosted, and then had a breakthrough infection with a variety of different variants. And then to the latest available vaccine at the time that we did the study, which was a bivalent booster vaccine. And so we compare how well 20 of the different naturally circulating SARS-CoV-2 variants, how well they're protected or not from these different exposure histories. So I'm just going to show you what one of these plots look like for one of these patients here. So you have patient from blood samples from a patient, and you can measure their neutralizing titers. So this is a proxy of how protected they are. Higher values means they have good protection. Low values means they are not very well protected. And this was a patient who had a wild-type convalescent infection. And so you can see they would have had high titers, good protection against the earlier strains, and then would have had low titers against the more later the strains that arose later. And so now this is just one patient. Imagine this data repeated 115 times. So one way that this type of data is represented is on an antigenic cartography. And really the intuition here is if 2 points are close together, it means they're antigenically similar. They tend to be neutralized by the same serotype. And if they're very far apart, it means they're antigenically dissimilar. So just to orient us, here I'm just plotting the natural variants of concern for SARS-CoV-2. They're colored, so each point here is a variant. They're colored by the date in which that variant emerged. So you can see that the earlier variant, the alpha, delta, gamma, Wuhan, are all clustering together in light pink. And then you move towards the Omicron variants, which are further away in darker orange, till the most recent variants, again, at the time that this study was done, which was HV.1 and JN.1 in darker red and further away. And so really the question that we wanted to ask is, can we use computational tools to map out what this trajectory would have looked like before it happened? So to do this, we developed a new method, which we called EVAX, for designing future mimicking proteins. So we combined the fitness predictions from the tools that I described earlier with 2 different components. And again, we included these 2 components because the sequences that the models were trained on were from spike sequences from prior to COVID-19. So these were spikes proteins from coronaviruses that did not infect humans, coronaviruses that infected other animals. And as the virus was moving, SARS-CoV-2 was spilling over and infecting humans, it was under different immune pressures and specifically escape from antibodies. And so we wanted to add these 2 components to factor that into account. And so the first one is accessibility to antibodies. So whether the residue that's being mutated is on the surface or the core of the protein, and then dissimilarity between the wild type and the mutant amino acid. So as species evolve, we often see mutations between amino acids that are physicochemically similar to each other. But in the case of being able to escape antibodies, you actually want mutations between amino acids that are dissimilar to each other in order to disrupt that surface and prevent the antibody from recognizing that surface anymore. So we combine these three metrics to get essentially an in silico deep mutational scan, where we have every position on the protein on the x-axis here and every possible amino acid on the y-axis. And then we look at the top 1% of most likely mutations, and then we score every possible double mutant in this top 1%, and then we combine those double mutants to get higher order spike constructs. These designed spike proteins then went into pseudovirus assays and were measured for infectivity. So how well does the designed spike protein bind to human ACE2 receptors and infect human cells, and how well is it neutralized by the different exposure histories that I showed you a few slides ago. So we repeated this at 5 different time points in the COVID-19 pandemic. So this is a tree that's meant to represent the different variants of concern and when they arose. And in triangles here are the protein sequences that we designed. So more specifically, what we did is we trained a model on all the data available up until, say, BA4 or 5 arose. We would use EVAX to generate spike constructs that we think mimic future evolution. And then we compare how well these designed constructs look like the real variants that emerged after. Now, before I show you the results for that, just a note to say that designing functional proteins is very hard. Anyone who's done any type of protein engineering, you know that if you take a protein and you start adding or changing mutations to it, chances are you're going to break the protein, right? There was the example we talked about earlier where you do just that simple swap and you end up going from 2 functional to 2 broken proteins. So in order to calibrate this, if you look at what percentage of all possible proteins that have 1 mutation, what percentage of them are viable or functional, that's about 70%. If you randomly add 2 mutations, that number drops to about 40%. If you add 3 mutations, that number drops very quickly to 20% and so on. And these are if you start adding mutations at random. Of course, we can be a bit more careful with how we combine mutations. So in this case, mutations were added that were seen very frequently in the pandemic. So there are mutations that are individually or in different combinations are functional. And if you start combining these individually functional mutations, you see that the decrease is not as severe, but it's still fairly drastic. If you compare that to our designed spike proteins, we see that we were able to retain functionality as we added more and more mutations. And in total, about 94% of our designed spike proteins were able to infect human cells. So that's great, but do they mimic the future evolution of the virus? In other words, do they map out this trajectory that we see in antigenic space for SARS-CoV-2? So now I'm showing you in triangles where these design constructs fall. And you can see that the designs on the Wuhan strain moved towards the gamma and alpha variants that emerged almost 12 months later. If you look at our designs on BA2.12.1, you see that they moved towards the BA4.5 variants that emerged 3 months later. Our designs on BA4.5 look like the BQ variants that emerged 8 months later, and so on and so forth at every time point in the pandemic. And so what this was telling us is not only can we predict how viruses evolve, but we can likely forecast how our immune system is going to respond to different changes before they happen. And so how can we use that then?
Sorry, Noor, can I ask a question right now?
Yes, please.
On these plots, you have antigenic distance on X and Y, so this is for different antigens, or what is it?
Yeah, so each point is a different antigen. that we measure neutralization across this panel here. And so essentially, this is just a dimensionality reduction. It's a multidimensional scaling. And the intuition is that it retains some form of distance where each box here is about a 2-fold change in neutralizing titers in either dimension.
Okay, thank you.
Yeah. Okay, great. So we can predict how viruses evolve. We can predict how our immune system is going to respond to them. How can we use this information to design better vaccines? And so one of the issues in the ways that vaccines are currently being made and produced is that we, whenever a new vaccine comes out, we evaluate how well it protects against current or previous variants of a virus, because that's all we had access to. But ideally, we'd also want to test against future variants, but we don't have access to those future variants. And so can we use our computationally designed proteins that predict what those variants are going to look like as a proxy? So we wanted to demonstrate this with one of the previous SARS-CoV-2 vaccines. So this is what we said is at the time that the bivalent booster vaccine was being developed, it was being evaluated on prior variants that had been circulating before. And one of the ways in which protection is measured is against these neutralization titers. So high values means the vaccine would give you good protection. Low values means the vaccine will give you low protection. And you can see that the bivalent booster would have given you very high titers against the variants that were circulating at the time. And it would say, great, this looks like a good vaccine option. We should move forward with it. But if you had access to our designed spike proteins that you could measure this bivalent booster against, you would see that there's a diversity of immune responses. And some of them are escaping quite severely from the bivalent booster vaccine. And so this would have indicated that the bivalent booster likely wouldn't have been effective for a very long time. Now we have the advantage of hindsight. We can go back and say, okay, well, we know which later variants arose. Where do they fall in this distribution? And we see that they fall within the predicted distribution of escape that we would have been able to anticipate with our computationally designed constructs. And so we have this panel of designed proteins for evaluating not just SARS-CoV-2 vaccines, but we also have one for seasonal flu vaccines, as well as for avian flu, and we have one for HIV that's in progress. But a panel like this can be used not just to evaluate how well vaccines are likely to continue to be protective in the future, but any type of antiviral, whether that's a small molecule drug or a therapeutic antibody. You can use these types of panels to give you an indication of what the future protection is likely to be given your particular antiviral of interest. Okay, but beyond just evaluating how well vaccines can or can't be future protective, can we use computational tools or machine learning to actually help us design better vaccines? And better in what way? There's two ways in which we are focused on. The first is broadening the immune response. So instead of a vaccine being protective against a small subset of viruses, can we try to broaden it and make it protective against a more diverse set of viruses? And then the second way in which we're focused on making better vaccines is, can we future-proof them? Make sure that they're protective not just today, but a year or two down the line. So I'm going to tell you two examples of vaccine designs that we have ongoing. The first is for a broadly protective vaccine. So there's a lot of interest in developing a pan-coronavirus vaccine, so a single vaccine that would protect us against a few different coronaviruses. And of course, we're most familiar, or a lot of us are very familiar with the SARS-CoV-2 coronavirus. So this was what caused the viral outbreak in 2019. But then there's been a lot of different spillover events from other coronaviruses over the last few decades. The earliest was from a rodent into cows and then eventually into humans, OC43. This is a human coronavirus. It's often associated with the common cold. We get mild symptoms. That spillover happened in the 1960s. And then there's been a few more. And then, of course, SARS-CoV-1 and MERS more recently. And I first heard about work for a pan-coronavirus vaccine on this great podcast, if anyone's interested in virology, called TWIV, This Week in Virology. And they were interviewing Pamela Bjorkman, who's a scientist at Caltech, and she had developed 2 different nanoparticle-based vaccines. So this is a newer vaccine technology where instead of more traditionally vaccines are attenuated or dead virus vaccines. Of course, the SARS-CoV-2, we had an mRNA vaccine. So this is a different type of vaccine modality where it's a protein nanoparticle. So it's essentially a protein cage that you can attach different proteins to at 80 different positions. And so the gray part here, that's the nanoparticle. And then in red here is the receptor binding domain, which is just part of the SARS-CoV-2 spike protein. And in this case, they had 2 versions of this vaccine, a homotypic, so one that was decorated with the SARS-CoV-2 receptor binding domain. And then they had a mosaic nanoparticle vaccine, which was decorated with different receptor binding domains from different coronaviruses. And what they showed was that this mosaic nanoparticle was able to protect across a diversity of different coronaviruses, not just the ones that were included in the vaccine. But what we thought was, well, what if we could, instead of relying on naturally circulating antigenic proteins, could we use computational tools to design which proteins we decorate these nanoparticles with? And so we have 10 vaccine candidates that are now in preclinical trials. We're seeing if the proteins express, if they can be attached to the nanoparticles. So I'm sad to say that we don't have any results yet to show for these. But hopefully in the next few months we'll have something to share. And the second vaccine that we're working on is for future proofing vaccines, and this is an avian flu influenza vaccine. And again, the idea here is for this particular vaccine, we're doing an mRNA where you immunize similar to SARS-CoV-2 with just the mRNA from one of the antigenic proteins. For SARS-CoV-2, the protein that was included was the spike protein, and the analogous protein in flu is the hemagglutinin protein. And what we wanted to know is, can we identify which regions on this protein are neutralizing epitopes, which means that if an antibody were to bind here, it would neutralize the virus or stop it from being able to infect human cells. And we'll find these neutralizing epitopes and then include future mutations that we think are likely to arise. That way, the vaccine would protect against those mutations as they occurred. And then the second thing with flu is we wanted to minimize or hide non-neutralizing epitopes. So these are regions where antibodies can bind, but won't neutralize or prevent the virus from infecting human cells. And so we wanted to hide or mask these by mutating these regions. So again, we have 12 different vaccines that are currently in small animal trials where we're immunizing mice. Sadly, the process of evaluating vaccines takes a long time, so we don't have any of the results here yet to show that these design vaccines can really future-proof the immune response, but hopefully we'll have more to show in the future. Okay, so just to summarize, I've told you about how our methods or computational methods can be used to forecast and predict how viruses evolve, how we can use that information not just to say which amino acid sequence are likely or unlikely to occur, but how our immune system responds to those, whether I will continue to be protected or not. And then ultimately how we're using both of those bits of information to design vaccines and therapeutics with an eye towards the future. I shared a lot of work, which was done with a ton of collaborators. All this work was done in Debora Marx's lab with the students that are highlighted in blue here, as well as experimental collaborators across different Boston institutions. Okay, and that is all. Take any questions.